Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody to In the Interim. This is a podcast of Barry Consultants on all things science of clinical trials, medical decision-making, drug development. And we are statisticians, typically talking about the science of this. Today, we have a really cool topic and a fun one for me, and I know a fun one for my guest. For the first time on In the Interim, my guest is Dr. Nicholas Barry, who also happens to be my son. And the topic for today is lessons for drug developers from the world of sports. So we're going to talk about what clinical trials, drug developers could learn from examples in the world of sports. So Nick, welcome to In the Interim.
Thanks. Yeah. Happy to be here. I'm happy to talk about this. I'm glad to be talking about sports because, a lot of our relationship when I was young was based on sports. You were my coach for, 15 years playing baseball, and a lot of how I started to perceive statistics was through sports, through watching Twins games, through watching baseball, through watching other sports, and sort of learning about all of the weird things that would happen and how a skeptical statistician like you perceives sort of home run races in the late 90s and batting titles and things like that. And so a lot of the way I learned to infer about statistics came from sports. So this is a sort of near and dear topic. And I wanted to be a sports statistician for a long time. Sort of when you were at Texas A&M, you wrote this column. The statistician reads the sports pages in chants. And so I would read those, I'd look at those, and sort of, it got me interested in that. And I sort of searched out a sports path too, right? I applied for some internships in sports. I worked with Hal Stern, who wrote a lot of sports stuff back in the day. And so I was on a statistics path for a while before I veered to clinical trials. So this is a perfect sort of merging of the two worlds for me.
Yep. Okay. So, and many of those experiences were my same experiences with my father as well, who's also a statistician and loves sports. And so we're going to talk about various concepts in sports. And this is the first of the first topic we're going to talk about. And we have multiple other topics that I think are really valuable for drug developers. And I think the examples will bring home some of the concepts. And so that's what we're also going to try to make clear here. We're not going to get too deep in the sports, not too deep in the drug development, but make sure we tie them together. So today's topic is regression to the mean. And it was talking about the family love of sports and the family love of statistics. If somebody in our family brings up an interesting thing that happened in the world and says to my mother, your grandmother, gee, what do you think that is? She will 90% just say regression to the mean, not necessarily knowing what it is, but knows that that's the answer to most of the questions it's regression to the mean. So what is regression to the mean? So we'll talk about it within sports, and then we'll talk about how what it means in the world of clinical trials and statistics and science. So my first experience in regression to the mean that I remember, it was the 1977 baseball season. I was 10 years old. I'm a little bit older than Nick, not surprisingly. And in 1977, being 10, I loved baseball and I could read the box scores. And I understood how to extrapolate statistics at one point in the season and say, what is going to be the end of the season within that? And the first of those was home runs. So George Foster was on my favorite team at the time, the Cincinnati Reds. The Minnesota Twins were terrible at the time, by the way. So the big red machine were the Cincinnati Reds. And George Foster, halfway through the season, had 31 home runs. And he was leading the league in home runs, and he had 31 home runs. And I was sophisticated enough to double that and say, okay, that's a pace of 62 home runs by the end of the season. At the time, the record was 61. The famous story of Roger Maris's 61 home runs. He beat Babe Ruth's record of 60 home runs, but that's the best that had ever been done in a season before that, and George Foster was on pace to break that. And I knew that halfway through the season, players have half of their expected number. And so he was on pace to break it. And as a 10-year-old, I thought he was going to break it. And I even mentioned this to my father, who's a statistician, said he's going to break Maris's record. And we made a wager based on that, where bet whether or not he would break the record. And I thought at the time my dad was nuts, that he would, not only would he make that bet, but he gave me 54 or more. He said, I'll bet he doesn't even hit 54 home runs by the end of the season. So at that point, does he hit another 23 home runs when he hit 31 halfway through the season? That seemed like this was a sucker bet for my father, that of course I'd make that bet.
Yeah.
And of course, he didn't hit 54. He didn't hit 62. He hit 52, which is actually a very good number. From his 31, he hit 20 more home runs, which is on pace. That 21 is a 42 home run hitter, which is an incredibly good home run hitter. So
sure,
he did really, really well, better than average, but not the 31 pace in that. Okay. And this happens baseball season after baseball season. A really fun thing about that season was my Minnesota Twins, Rod Carew was flirting with batting 400, a batting average of 400, which is the number of hits divided by the number of attempts. And that's easy to project. It's the batting average. And at that point in the season, he was hitting over 500. And so we might, in this 1977 season, have a 400 hitter and break the home run record all in one season on my two favorite teams at the time.
He was hitting over 400.
He was hitting over 400.
Yeah, which is an astronomical number. Like, this has happened less than, a handful of times in history of the game.
The last time before 1977 that it had happened, which is still the last time, it has not happened even since 1977, was Ted Williams hit 406 in 1941. And so this has become a number that baseball fans know this hasn't happened. And so that's a very rare thing. And he had the best batting average in the league at the time. And I didn't make a wager on this, but he ended up hitting 388 actually, which is one of the highest numbers since 1941, an incredibly high number. Rod Carew's a Hall of Fame baseball player because of his batting average, incredibly high number. And we've had several people since then that have done similar, flirting with that 400 number. George Brett did, actually Lenny Dykstra did, Tony Gwynn, even Joe Mauer flirted with it in another Minnesota Twins, and nobody has accomplished that. So this is something we're very familiar with in sports, of this kind of phenomenon happening. So what does it mean in that scenario? Was Rod Carew truly a 400 hitter? And was George Foster truly a 62 home run hitter at that point?
Yeah.
And they weren't within that scenario, and they were the extremes. And so if we were to say, how good are they really? We would never estimate them. And my father knew that George Foster was not a 31 home run hitter, which is why he was very comfortable betting. He knew he probably wasn't even a 21 home run or a 23 home run hitter, which is why he took that bet, that's an extreme number. His estimate was regressed. Now, George Foster had previous performances, but also regressed towards the middle in that setting.
I was going to say, I'm sure that Don, your dad, looked up George Foster's previous 3 years and saw he hit 35 home runs to 40 home runs every year and made a deduction.
Yep. So we'll come back to sort of how we might estimate what they really are. But this phenomenon happens in every sport. It happens in hockey, it happens in baseball, it happens in basketball. We have people and teams. We have teams halfway through a season that are on pace to break the record number of wins in basketball. We've had some do that, and they fall short of that. And we always talk about that. We've actually recently had teams break, and we might have a team this season break the record for most losses in a season. These extreme sort of things that we've talked about, and then it doesn't happen. And in the sports world, we hear reasons why that happens. We hear the pressure, the scrutiny of the media, everybody asking about it day in and day out. It's really hard to continue such a pace.
Yeah, they got in their heads, and the whole.
Yep. So let me provide another example of this in a sport Nick and I both love, which is golf. We play golf, and if you're looking on video, we have golf shirts on. Within this, we love to play golf. So let me provide another example. A money-making gig for us potentially is a golf tournament. And I'm going to quote numbers from the 2017 U.S. Open, but this happens week in, week out, very, very similar numbers within it. The U.S. Open might be a little bit more extreme because there's more of a variety of players in the U.S. Open than the run-of-the-mill weekly PGA Tour tournament. So in the 2017 U.S. Open, if I take the baseline score, and I'm calling it baseline, but we're going to start talking about clinical trials, but the first day, what the golfers shot on their first day in the U.S. Open. And I'm going to break them up into four groups, four quantiles. The top 25% of golfers, the second 25% of what they shot on day one, the third and the fourth, the bottom in terms of the worst score within that. So I'm going to take those golfers, and let's talk about the worst quantile at first. The worst quartile of this, the 25% that shot the worst score. They shot an average of 78 on day one. U.S. Open's traditionally very, very hard, and this round was a hard round. The average was 78. I'm going to do an intervention on that group. I'm going to go around to each one of them, and I'm going to think really good thoughts about them, and I'm going to give them encouragement in that. And so what happened to them on day two, when I intervened on them, is they averaged 3.04 shots better on day two than they did day one. Their change from baseline from day one to day two was 3 shots better. I did that. I caused them to shoot 3 shots better. And does 3 shots matter in a PGA Tour tournament? 3 shots around is multi-millions of dollars.
Yeah.
It's the difference between being on one tour or another tour, winning major golf tournaments. It's millions of dollars. It's an enormous number in that. And think about a 4-round golf tournament, that's 12 shots. If I could bottle that and have a 3-shot effect, I'm making millions in this. Okay. Likewise, the golfers that shot in the top, the best quartile, they got 3 shots worse on day two on average. I didn't think good shots of them, and in fact, I thought bad thoughts about them, and they do 3 shots worse on day two. This is repeatable. This happens every single week in the PGA Tour. This happens all the time in the circumstance. So what does it mean? Did my thinking about them affect them in any way, shape, or form? No, certainly not. in that. That doesn't mean other things are not understood whether they had an effect. But
the bottom quartile of people didn't feel no pressure and go straight at pins because they had a tough first day. It wasn't some conscious, concerted effort by the players to do better, play freer, right? This is variability. It's randomness that led to it.
So announcers will tell you that I had a bad day and I was so super aggressive and I wasn't worried about my positioning, and you shoot much better when you do that. Or vice versa. Now you're near the lead. You didn't sleep well. You kept thinking about hosting the trophy and you shot worse with it. So people attach reasons to that something that is nothing other than randomness. And this is nothing other than randomness.
And this isn't just like casual fans. I mean, the players themselves say this, announcers that played for 15 years say this. And this is a, people who have spent their life playing the game still contribute a lot of, variability to physical aspects rather than the fact that there is just a lot of variability in day-to-day scores.
So we have this term regression to the mean. What do we mean by regression to the mean within a setting like this? There's the average score on day one, which I think was about 73.5, something like that. And we have the average of those golfers. All these golfers are participants, and the average is 73.5. And somebody shoots 78 on day one. And let's ignore previous tournaments that they come into the U.S. Open. We're interested in that. If you estimate that individual's true average score on that golf course is 78, you're shocked when you find out the average of the people in that quartile got better by 3 shots on day 2. And there must be a reason for that. You could think everybody is a 73.5 and all golfers are identical and there's no difference between them. You'd estimate those golfers to shoot 73 and shoot 5 shots better on day 2. Not all golfers are all the same. They're different. Some golfers are better than the other ones. We know that. And especially the U.S. Open, there's more heterogeneity in those players. There's variability across players.
Yep.
There's variability on a day score within a setting. So what does it mean if somebody shoots 78? They've shot a high number. They're probably not as good as the average, but they're not as extreme as the 78. If we were estimating them statistically, we would take an average of their 78, their score, and the average of all the golfers, if this is all we knew trying to estimate them, which is 73, and we would do some estimate midway between that.
Yeah. And how far you go from one to the other depends how well you think you actually know how good golfers are, and how maybe if a golf round was 1,000 holes, you think you know more about the player. So the amount of data you have and the amount of belief you have in how good the players are goes into this weighting of where you are between the two.
Right. So in only using the 2017 U.S. Open, the two key things about how much regression you would do towards the mean is the between subject variability, how variable are golfers, and then the within subject variability in an 18-hole score. And by the way, I've spent some time in this. It's a little less than 3 is a standard deviation of within a professional golfer on any one day. Within that, the between golfer variability in this U.S. Open is quite a bit bigger than that within the setting. The bottom quantile was 78. The top was 69. There's probably a 5 or 6 standard deviation across golfers. So we would use those in that setting. Now, to provide that estimate of that. So we're not at all surprised when the next day scores for all of them shrink in towards the mean. It's inevitable because the first day score variability pushes them outward. And their truth is somewhere in the middle of that. Variability always goes outward. That's what variability does. So we would always say when somebody does better on day 2, they go from 78 to 75, we say, oh, that's regression to the mean. Our estimate, if I was betting on somebody that shot 78 on day 1, I would probably estimate 75 on day 2. And I would bet and I would behave that way in the particular setting like that. Meanwhile, somebody that shot 67, I would probably guess 70 or 71. It's a regression to the mean. And statistically, a common way we do this in Bayesian approach is something called hierarchical modeling. And the hierarchies are across players is a hierarchy, and within players, their different scores is a hierarchy. And that's used to provide these estimates. And we do it in sports all the time. We also do it in drug development. Now, this is a natural phenomenon. And this is a natural phenomenon if you're rolling dice. This is a natural phenomenon if you're flipping coins, if you're playing golf, if you're playing baseball, if teams are having outcomes. It's pretty well understood in sports. It's sometimes understood in drug development. And this is the part that drives us nuts, is people attach reasons to it that are not right. They're wrong. And then they behave that way to it, which creates bad decision making, bad betting, bad decision making, bad paying of baseball players, expecting them to do the same. And there are some you may heard of. So there's something I don't even know if people talk about anymore. The Sports Illustrated jinx. Everybody recognized it, that players or people or teams got on the cover of Sports Illustrated, a weekly magazine. And what happened is they tended to do worse. And people referred to it as a jinx of being on Sports Illustrated.
Sports Illustrated doesn't exist anymore, but it's now the Madden jinx or whatever, the video game that comes out. You're on the cover of Madden and then the next year you do worse. But it also exists. Crazy coincidence that there's also a Madden jinx, like a Sports Illustrated jinx.
And people believe that's a real thing. The jinx is a real thing. You've heard the sophomore jinx. People have a great freshman season. They don't do quite as well in their sophomore year, and it's a jinx.
Yeah, sophomore.
We don't hear this anymore because if you have a great freshman year, you go pro. There's a rookie of the year jinx. If you go look at players who win rookie of the year in baseball, in any sport, they do worse the next year. They're complacent. It's a jinx. And it's nothing but regression to the mean, largely.
They overperformed in their first year.
Yeah. And if you believe they're going to repeat that performance, that's the mistake you made. And then you think, okay, we have to attach an effect to this within it. Media pressure, scrutiny, team chemistry. Oh, the team chemistry, that great was seasoned. The next year the chemistry wasn't very good, which means we don't know how to explain it, so we invent chemistry for it. This happens in all kinds of things. Golf equipment. Who goes, you try a new putter when you're not playing well, a new driver, and you're doing better.
Yeah.
Golf teachers benefit from this greatly. Who goes to see a golf professional? Somebody who's struggling. I'm on that left side. I'm shooting the higher scores, and I play better. And I want to pay more money to that golf professional in that setting. Now, lots of people benefit from this. Chiropractors benefit. Your back hurts, you go in. Doctors benefit. I'm not feeling well, and then I feel better in the setting. And we attribute it to that. Rehab, therapists. We use trinkets. We put magnetic bracelets on our arms to make pain go away, and it goes away, and this sort of thing. So there's all kinds of this shows up in every walk of life. Now let's talk about this in clinical trials. Yeah,
you've been really pessimistic. Like everything is fake. That's not necessarily what you're saying. You're just saying that every time you observe something extraordinary, especially in the circumstance where the population that underwent this procedure or something like that was different than the normal population, but you're just saying shrink back the results. You're not saying that it's fake, golf teachers don't work. It's just that if you see immediate benefits after you go see a golf teacher, some of that benefit is due to regression to the mean. There are benefits of some of these things, but believing at face value the change you see right away is naive when we know that there's going to be some regression.
Yeah, exactly. And doctors are phenomenally important. I'm not saying don't go to a doctor.
I know.
For
Exactly.
example.
Yeah, exactly.
Yeah. Your uncle and my brother is a golf professional. Yeah. So they absolutely play a role, and they will help you shoot better scores. No question about it. But
it's hard to figure out how much of it goes to each thing. Like, it's really hard to assign how much it should shrink.
Yep. Okay, so let's talk about this in clinical trials and drug development. Yep. And now you can start to imagine some of these effects showing up within it. A very one that I think is very misunderstood is something called the placebo effect. And people talk about clinical trials, that in a clinical trial, and when people say the placebo effect, what they mean is somebody takes a treatment that is inert, but they're taking a treatment, and the act of taking that makes them perform better. And you can see where I'm going with this. I was labeling my thinking about golfers in that situation as kind of a placebo. And the golfer doesn't know I'm thinking about them in that setting, and they do better. So let's take a clinical trial, and what happens in most clinical trials is people who enter a clinical trial are like that golfer who shot 78. They're doing poorly relatively on their scale. And it could be high blood pressure, it could be high weight, it could be pain. I'm not sleeping well in that scenario. And this happens in Alzheimer's trials, that people that their memory has been particularly bad, I'm doing worse. They enter in trials, and in many of those scenarios, patients who get no treatment or an inert treatment, they do better. And everybody calls this a placebo effect. Now, in many circumstances, that's the exact same thing we talked about. It's regression to the mean. Now, there are certainly some cases where I believe that the act of taking an intervention may help. And you could imagine sleep. You could imagine potentially pain trials is where they've talked about it. And there's actually been trials where they give placebo or they give nothing. And there's a difference between those. But most clinical trials, this is what happens. And
placebo effect's interesting. It's like you take statistics in high school and you don't know anything about statistics, but they're teaching you about placebo effects, and it's like a immediate thing. And so you just assign sort of improvement without getting an active drug to the placebo effect, without assessing the population or thinking about sort of populations going in. It's just, getting the drug makes you think you're going to get better, so you get better. In the placebo, getting a placebo makes you think you're going to get better, so you get better, which is usually not what's happening.
And so the misunderstanding of this has an effect. And let me give you an example that happened to me a couple of weeks. A company collected single-arm data. And what that means is they don't have a placebo, that they run a trial where everybody gets the treatment. And they have a baseline marker of severity. And by the way, that's the outcome measure, but that's also measured at baseline. It's similar to their first round in a golf tournament within the setting. And they collected it across a wide range of patients, and they noticed, wow, patients that have higher baseline, they improved more when we gave them our drug. Now, In the golf example, that's taking that top quartile and noticing they regressed 3 points in better. By the way, the people in that golf tournament in the second to the worst quartile got better by 1 shot.
No.
The data from this company looks almost identical to the golf tournament. Remember, in a clinical trial, we record your baseline before you get the intervention and what's your score. There's variability in those. There's variability across time. A single patient goes up and down on almost every measure: weight, blood pressure, cognitive scores. This cardiovascular endpoint they were looking at goes up and down within a patient over time. And so they're now running a randomized trial in that particular population. They're going to run a randomized trial. So we get to find out, do the placebo get better? And I told them, you're going to get a very large placebo effect in your trial. And they looked at me like, what are you talking about? And their perception was what the investigator tells them about how good the drug is, or what we know about it. All those things are what cause the placebo effect, and not the inclusion-exclusion criteria at the beginning of the trial, which causes a huge amount of regression to the mean, which we don't understand, and we call the placebo effect.
Yeah. The placebo effect's not a psychological thing that messes with patients. It's a statistical process that you just described, right?
So the regression to the mean is the statistical process that we confuse for placebo effect, which in some cases, placebo effect is a real thing. In that sense, we don't understand very well which is which. We don't understand very well at all which is which within that setting. So they made huge drug development decisions to enroll that particular population from a single-arm trial without a control, not understanding that, by the way, the people on the good side might actually be the ones that have the bigger benefit relative to a control. And now they're jumping into a very, very large trial. Do they understand this particular effect? Or are they making a bad decision? The beauty of it is the randomized trial is going to tell them, and we're going to figure that out. But do they understand it enough to make good decisions at this point? Other examples that show up that regression to the mean is critically important. We run a lot of trials and we look at subgroups of patients. We have a trial design now called a basket trial. And the baskets in a basket trial they're different kinds of patients. And a very common one of these is we have a treatment for cancer, and we enroll different types of cancer. It might be head and neck cancer, breast cancer, lung cancer, GI cancer, all of these different types, or they may be subsets of kinds of cancer. And we run trials in them, and we go into 8 different types, and we find out, these 2 types we had the best effect in. And we look at the estimate from the 2 best out of 8, and we run a trial after that, or we try to estimate the effect in the 8th, the best one of the 8, and the data were the number of responses we got. Suppose we're looking at a cancer trial and we look at how many patients responded, and we got 10 out of 20 patients in the best responded. And all the other types had worse responses than 50%. I, as a statistician, don't believe the response rate's 50% in that one. It's exactly the same as the golfer that shot 78. I don't believe they're 78. They're better than that. This 10 out of 20, I would estimate their response rate to be closer to the mean response across all tumor types, depending on the variability of that response and the variability of 10 out of 20, which as statisticians we understand.
Yeah. And your hierarchical model, like we talked about with the golf example, now says there is some variability across the cancer types, and we know that it might actually work better in some, but there's also a lot of variability in how many responses you're going to observe, about 20 on 20 subjects. And so 20 is not that many. We don't know a lot about how well each of these cancers or how bad each of these cancers are. So we shrink back and we say a lot of the variability in this was due to there only being 20 patients per cancer type. So just pull everything back towards the middle. And if you're predicting what's going to happen in phase 3, you're not going to predict 50%. And if you do, you're going to cost yourself, millions of dollars potentially. So you just shrink it all the way back really close to that mean. Probably if you only have 20 subjects, you probably don't vary very much from the overall mean of all the types.
Yep. We see lots of this happening where we have units in the case like this. We have trials with multiple arms, many doses. And the effect on one dose, you should never estimate the effect on one dose without using the other doses. It could be a hierarchical model. It could be a dose response model. We have endpoints. We collect a lot of endpoints in clinical trials, and we analyze 12 endpoints in a particular disease where we think a treatment may have an effect. And this one did the best out of the 12. And this is like the 1977, halfway through the season, we see this effect. Now we're going to run the rest of the season. Do I think that effect in the 12th endpoint is going to continue at the effect I see, or is it going to shrink towards the other 11 endpoints within that? It's very similar scenarios to sports where it seems almost obvious in that setting. But a lot of these things aren't naturally done in clinical trials. Publications provide this single estimate, and it's up to the consumer to do that themselves, which I think experienced people in this industry do that, and they understand that. There's this whole, it's almost controversy about the failure for phase 2 trials to replicate in phase 3. And a lot of the things we just talked about are reasons why that doesn't happen. But the phase 2 process itself is relatively small sample sizes, so the variability in that measurement, it can be large. And we run hundreds of phase 2 trials. What phase 3 trials do we run? Those ones that are doing really well. And there's a natural part to this, and there's many phase 2 trials. And you see the extreme right tail of those that do well. They run phase 3. Lo and behold, the effect is not reproduced in phase 3. Sometimes it's completely gone, but sometimes it's just smaller. It's regressed into the average of the effect. And people think that this is a controversy. This isn't reproducible science. What's wrong with what we're doing? It's regression to the mean.
A huge problem with that.
As your grandmother would say, it's regression to the mean.
A huge problem with that, though, is that now we're powering our phase 3 studies based on the effect observed on the best dose in phase 2. And, if we're regressing our estimate, the truth is probably not as good as that. You have an underpowered study. And when you inevitably observe, 80% of the phase 2 effect in phase 3, you have a p-value of 0.07 or something like that. And so it's not just our estimate was off. I mean, this can have huge implications in this sort of life cycle of a drug.
Yep. So I hope there were lessons there, but let me turn it back to you, Nick, because I know this is a topic we talk about with a lot of people. And your friends heard this topic, and they're in personal finance.
Yeah.
And what does the regression to the mean to them? So what would you say to people outside of drug development, outside of sports? What does regression to the mean mean to somebody who's doing personal finance and buying stocks?
Yeah, I think the general lesson that I would say to this sort of lay person is that anytime you observe something extraordinary and you get something that's you've never seen before, like this is an amazing result, you just smooth over that and you realize that there were maybe a lot of opportunities from other places to observe this amazing result. There's a lot of stocks that could have performed really well. And when you start to base decisions off of recent amazing things that happened, you're inevitably setting yourself up to be disappointed if you expect that to be reproduced and to happen over again. So when you observe something amazing, just smooth over it. Realize that it might be good, but it's probably not actually a world beater in that case. And so just temper your expectations every time you start to make predictions based on past data.
So if there are 50 stockbrokers in a company, and in a particular year, one person is the best stockbroker, and they earn a certain percent, I don't even know what the best would be, 30% on their investments over a course of a year. And then the next year they don't do 30%.
Yeah.
Were they lazy? Yeah. Were they lazy? The media scrutiny got to them.
Yeah. Exactly.
It's that they actually were never that good, and the data got them there through randomness. Now they may be better than average. How much better depends on the variability in stock pickers and the variability in a particular year. So the lessons are in drug development. The lessons are in sports, in picking stocks. So this was our lessons learned for drug developers from the world of sports. Episode 1.
We'll see you later.
Yeah, we have more to come. So Nick, thanks for joining me. And
Thanks for having me.
We are in the interim.